All right, sorry, I lied. It said we only had, no, we didn't have any more sections for chapter 10 with muscle tissue, but we did have another one here, talking about cardiac muscle tissue and finalizing our muscle tissue chapter here. Learning objective, our primary one for cardiac muscle tissue, is to list and describe the similarities and differences between skeletal muscle and cardiac muscle. With cardiac muscle cells, they're short branching fibers as opposed to cylindrical fibers like skeletal muscle fibers. They have one or two nuclei, except as opposed to the multinucleated fibers of skeletal muscle. They're striated like skeletal muscle, that contain sarcomeres. They have many mitochondria, use aerobic respiration, and that's dependent upon the skeletal muscle fiber. They have intercalated discs, which muscle fibers do not have, that join the ends of neighboring fibers. These discs contain desmosomes and gap junctions, and information and a potential action potential essentially goes across these intercalated discs. Contractions are started by the heart's autorhythmic pacemaker cells, sinoatrial node. The heart rate and contraction force is influenced by the autonomic nervous system. It's not something we have voluntary control over. And then, of course, looking at it a little more closely here, we have the endomysium layer here outside the skeletal, cardiac muscle cell here, muscle fiber. And basically, look at this little portion of the pericardium here. We have our gap junctions here, desmosomes, mitochondria in there, a sarcolemma of this muscle fiber here, a nucleus here, and cardiac. This is the whole cardiac muscle cell here. So what did you learn? There are three anatomic or physiologic differences. What are they between the skeletal muscle and the cardiac muscle? And finally, talking about our smooth muscle tissue a little bit here, you should be able to identify the organs of various body systems where smooth muscle is located. Compare the microscopic anatomy of smooth muscle to skeletal muscle. Okay, explain the sequence of steps in smooth muscle contraction. Briefly explain the different means of controlling smooth muscle. Explain the primary functional difference between multi-unit and single-unit smooth muscle. Compare the location regulation of both multi-unit and single-unit smooth muscle. That said, smooth muscle is located or found in a variety of organ systems with a variety of roles. Blood vessels of the cardiovascular system helps regulate the blood pressure and flow, and you'd also argue resistance, blood pressure, flow, and resistance. In bronchioles, the respiratory system, it controls the airflow to the alveoli and the lungs. In the intestines, the digestive system, it mixes and propels materials. In the ureters of the urinary system, it propels the urine from the kidneys to the bladder. And in the uterus, a female reproductive system. And of course, here we can see now some locations here. So we have smooth muscle here, the cardiovascular system, the blood vessels, right? And we have the respiratory system, the lungs here, and then we have a smooth muscle here, the lumen here, the bronchiole. And showing here the lumen again here, now referring to the digestive system with the large intestine and a smooth muscle. The urinary system, the urinary bladder in this case, showing the smooth muscle here, and the smooth muscle of the reproductive system in the uterus, the female. Our smooth muscle cells have a fusiform shape. It's wide in the middle with tapered ends, right? It's smaller than skeletal muscle fibers. Then the sarcolemma has a varied types of calcium channels, gated by chemicals and voltage, whatnot. Transverse tubules are absent. They're present in skeletal muscle fibers. The surface area is increased by caveolae, which are flask-like invaginations. Sarcoplasmic reticulum is sparse, unlike that of the basically of a skeletal muscle. And the outside of the cell is an important source of calcium. So you can see here, looking at our smooth muscle, we have the sarcolemma, and then we have our caveolae, little invaginations there, the nucleus, the mitochondrion. We have our cytoskeleton intermediate filaments with that smooth muscle cell here. Some dense plaque here, and showing that dense plaque. Actually, I'll get to the contracted smooth muscle cell in a second here. A little more broader view here, the dense body and the intermediate filament here. The green you can see there, our thick filament here, and our thin filament for the contractile capabilities of that smooth muscle. Basically, we're showing that here, showing that contracted smooth muscle. Basically, a contraction. See how it's shortened across there, more broad if it's relaxed. And then you can see a contracted smooth muscle here, and then relaxed, right? It can be obviously more lengthened when they're relaxed and shortened when they're contracting. The arrangement of anchoring proteins and contractile proteins of smooth muscle. The cytoskeleton is composed of extensive intermediate filaments. the smooth muscle. The dense bodies are the points where the intermediate filaments are interacting within the sarcoplasm. The dense plaques is where the intermediate filaments attach on the inner sarcolemma. The contractile proteins are arranged between dense bodies and dense plaques. They're oriented at oblique angles to the longitudinal axis of the cell, right? Kind of an angle there. And then the contraction causes a twisting motion, right? And of course, these smooth muscle cells lack sarcomeres and Z-discs. It's smooth, there are no striations or striations. Now, like skeletal muscle, smooth muscle filaments have actin, myosin, and troponin. Unlike skeletal muscle, smooth muscle filaments have myosin heads along their entire length and can form additional cross bridges. Filaments can perform the latch bridge mechanism, where the myosin attaches to the actin for extended time without using extra ATP, which makes sense. We're talking about muscles. Smooth muscle has to be active a lot more on a continuous basis, more so than most skeletal muscle would. Has calmodulin, which is the protein that binds calcium to trigger contraction, which you find that other skeletal muscle as well. Has myosin light chain kinase, which is an enzyme that phosphorylates myosin heads when activated by calmodulin. It has myosin light chain phosphatase, which is an enzyme that dephosphorylates myosin head. It's required for relaxation. So here we have with our smooth muscle, the opening of the voltage-gated calcium channels, right? Whereby the action potential fires and triggers an opening of the voltage-gated calcium channels. Calcium enters the sarcoplasm, primarily from this interstitial fluid out here. And then the binding of the calcium to the calmodulin here. And then activation of the myosin light kinase, MLCK, where the calcium calmodulin complex activates MLCK. It's a phosphorylating enzyme, right? Let's go from 3 here. And then activation of the myosin heads. These are myosin heads here, inorganic phosphate here, ATP breaking off ADP, inorganic phosphate, like we'd see with skeletal muscle, that reaction there, generally speaking. And this activated MLCK then phosphorylates, which adds a phosphate to the myosin head and activates the myosin, a relatively slow process here, which makes sense, right? We don't want necessarily overly hyperactive smooth muscle, at least generally speaking. So that cross-bridge formation of the power stroke, the reattachment, then these activated myosin heads bind to these thin filaments to form the cross bridges, and the myosin ATPase hydrolyzes ATP, not hydrolyzes ATP, provides the energy for the power stroke. This process is repeated. The force is generated and then transferred to the anchoring filaments, and the smooth muscle cell shortens. So the steps of contraction for smooth muscle: stimulus leads to the opening of the voltage-gated calcium channels. The calcium enters the sarcoplasm and binds to calmodulin, and this calcium calmodulin complex binds to myosin light chain kinase, MLCK, and this MLCK phosphorylates the myosin head. The cross bridges form. They pull on the actin, similar to skeletal muscle, but more slowly. And as thin filament slides, it pulls on these dense bodies. These dense bodies are attached to intermediate filaments, which are attached to dense plaques in the sarcolemma. These filaments move inward, and then the entire cell shortens. The relaxation of smooth muscle, cessation of stimulation, where there's removal of calcium from the sarcoplasm, the dephosphorylation of myosin by myosin light chain phosphatase, and it can be slow to relax due to the latch bridge mechanism. The smooth muscle contraction characteristics: there's a long latent period where it takes time for the phosphorylation of the myosin head there to slow ATPase activity. Long duration of the contraction period, slow calcium pumps, need for dephosphorylation of the myosin head, and a latch bridge mechanism which keeps us going. The slowness fits its functional requirements though, right? Its extended contractions maintain continuous tone, which is important. Continuous activity, in other words. In the walls of the blood vessels being a great example. Blood's got to keep moving. If blood completely ceases, well, that's the end of us, or the end of at least a component of us, which will ultimately lead to the nth degree that we don't want. Smooth muscle contraction characteristics are we have fatigue resistance. Energy requirements are low compared to skeletal muscle. It can maintain contraction without ATP through the latch bridge mechanism. So latch bridge mechanism is going to keep showing up here for smooth muscle. You can bet that's a concept that'll show up on an exam. The broad length and tension curve, which lacks limitations because there are no Z-discs. The myosin heads are present in the center of thick filaments and can contract forcefully even when 50 to 200% of its resting length. It allows the emptying of the bladder regardless of the amount of urine. As far as control of smooth muscle, the autonomic nervous system secretes transmitters with the muscle's response dependent upon the neurotransmitter present and the muscle's receptor for it. Smooth muscle, the bronchioles contract in response to acetylcholine, and it relaxes in response to norepinephrine, right? We'll talk about epinephrine, norepinephrine response more in nervous system a lot more in a later unit. Response to stretch, the myogenic response is contraction in reaction to stretch. The stress relaxation response is relaxation without relaxation after the prolonged stretch. Other stimulating factors include various hormones, low pH, low oxygen availability, high carbon dioxide, certain drugs, pacemaker cells, all play a role in stimulation or lack thereof. Two categories of smooth muscle are the multi-unit and the single unit. The multi-unit smooth muscle is arranged in units that receive stimulation to contract individually. It's found in the iris and the ciliary muscles of the eye, the erector pili muscles in the skin, and in the dermis, and the larger air passageways in the respiratory system, and the walls of larger arteries. And the degree of contraction depends upon the number of motor units activated and very much similar to skeletal muscle. Our single unit smooth muscle, the visceral, is the most common type. It's stimulated to contract in unison as cells are linked by gap junctions. It forms 2 or 3 sheets in the wall of a hollow organ and includes the walls of the digestive system, the urinary system, reproductive tracts, portions of the respiratory tract, and most blood vessels. That's what you're most commonly going to see in these single unit smooth muscles. As far as stimulation of single unit smooth muscle, it occurs through varicosities, swellings of autonomic neurons. These varicosities contain synaptic vessels with one type of neurotransmitter, either acetylcholine or norepinephrine. And the receptors are scattered across the sarcolemma, and they're through diffuse junctions. And the numerous smooth muscle cells are stimulated simultaneously, and stimulation is spread from cell to cell via gap junctions, so contraction is synchronous. That's important. So we have our synaptic vesicle here. I don't think it's necessary to zoom in here. Our autonomic motor neuron here, and varicosity here. Okay, the vesicle is being on that varicosity. And you can see that it's being the broader picture of what we see here for that smooth muscle cell. And we have gap junctions between them. And you can see the varicosity here again, an autonomic motor neuron, once again, there. And of course, here are synapses too, as well. So where is smooth muscle located in the human body? How are anchoring proteins and contractile proteins in smooth muscle cells arranged? What's the specific role of the following structures within smooth muscle cells? Calmodulin, myosin light chain kinase, and myosin light chain phosphatase. And what are the steps of smooth muscle contraction? What unique characteristics of smooth muscle allow it to fulfill its functions? Should be able to explain that. What are the various forms of stimulation for controlling smooth muscle? Explain the stress relaxation response of smooth muscle. And explain why smooth muscle of the eye is a multi-unit smooth muscle and the smooth muscle in the wall digestive organs is single unit smooth muscle. And that's the end of our chapter 10 on muscle tissue.